Hello, everyone. It's my pleasure to tell you a little bit about biometrical extension of latent growth curve modeling. As you can see, my name is Michael Neal. I'm a distinguished professor at Virginia Commonwealth University and always enjoy teaching the NIMH-funded Boulder, Colorado workshops on statistical genetics. So, we had the original latent growth curve model. This is shown here with three components of growth. That is a level component, which varies in the initial level that somebody has of a particular trait. There's a slope component, which would give a varying degree of acceleration or change. Could be up or down in growth over time, and a quadratic component that would influence the amount of curvature experienced by people over time. So this model is set up as a between individuals type of model, meaning that we'll have a sample size of many individuals each with up to 5 occasions of measurement. And the model is specified here for 5 time points. And note that in general, these latent growth curve models will require one more time point than the number of growth components involved. So if you've just got level and slope, you are still going to need 3 time points of measurement. So how do we convert that extremely well described latent growth curve model? It was very well described by Bobby Mulder in the earlier videos in this section. And extension to data collected from relatives is actually a remarkably straightforward kind of thing. First of all we really are going to do is make the random components of this model covary between relatives. And if you only had one class of relatives, say SID pairs or something, it would be perfectly okay not to attempt a separation of variance components into shared and non-shared within the family, but simply to estimate their covariance. So we could estimate the covariance between the level factors of SID pairs or between the slope factors of SID pairs, but we would also get information on level slope covariance within an individual, and we'd also get some information about level slope covariance from across individuals. Finally, one more thing to note about this path diagram is that at the bottom, there is residual variance specific to each time point. And quite often that residual variance specific to each time point can be constrained to be equal across time points. So what is labeled as separate parameters R1 through R5 here is more typically just estimated as a single estimate of unreliability of measurement that is the same proportion or the same amount of variance added to each occasion. In fact, it may be adding a different proportion of variance to each occasion because this model does not predict that the variance stays the same across the times of measurement. And indeed, given positive genetic variance due to A, C, or E, or any other source on a slope component, that is going to predict an increase in variance over time. It will also predict increases in covariance over time between relatives, and it may or may not show an increase in resemblance between relatives over time. And it is these parts of the data really that the model is trying to accommodate at all the time when it is fitting to a population-based set of data. Okay, so that latent growth curve model, here we've partitioned it and allowed the additive genetic common environment and specific environment components of level and slope to co-vary across relatives. What may also be necessary in this model specification is to allow for some covariance between those level and slope components. This is enabled in the diagram here as just a raw correlation between level and slope, but a more conventional approach would be to shift that covariance between level and slope up to the factor level as well, so that we can then partition the covariance between level and slope into additive genetic, common and specific environmental components of the covariance between level and slope. However, it's often important to realize that the covariance between level and slope is very directly a function of the age at which the data are centered. How the data are centered is going to change, not the estimates of the variance components themselves, but it will change the estimate of the covariance between level, slope, and any other factors that you may have, quadratic, etc. Here is a slightly simplified version of the model, and put that in quotation marks at the top right. But there's one thing that we've changed between this version of the model and the predecessor here, or even this predecessor, which includes a quadratic component. So in these two figures, here and here, you'll notice that the factor loadings are specified as fixed values. That is, they are not free parameters to be estimated during optimization. And in this particular model, that is similar, except that those factor loadings have been replaced by the actual age at assessment of each of the individuals in the sample. So here we have a classical use of definition variables, which enable a different model to be specified potentially for each different row of data, each set of independent observations, potentially quite differently. By putting the actual age at assessment for each of the occasions of assessment onto the path in the path diagram. And for the quadratic part, we simply square those ages at assessment and stick those on the paths from the quadratic factor to the observed variables. So in essence, for every twin pair in the sample, the model could be different because they were assessed at not exactly the same age. In some longitudinal data sets, you may find it difficult to obtain the actual ages at which people were assessed for reasons of confidentiality. But at least some kind of precise interval between the ages at assessment is going to be required, as well as one age at which people were assessed. So it can represent a little bit of a data security challenge for analyzing such data, that precise ages at assessment are sometimes not available. So the main features of the biometrical latent growth curve model is that it does allow modeling of longitudinal data, and it does so kind of regardless of the number of occasions that you have. That is to say that if we had 100 measurements for an individual, it would be quite feasible to fit a latent growth curve model. And the reason it would be quite feasible is because of the very sparse parameterization of the model. It's got relatively few free parameters. There's, particularly if you accept that all the residual measurement errors are going to be the same. That's one parameter. And then you've got parameters for variance of level, variance of slope, and variance of quadratic, and the same kind of thing for the means of those. And you also have to allow for covariances between those latent growth factors. But beyond that, there isn't anything that means that the number of parameters is going to increase with the number of occasions of measurement. So it's very efficient, and it can result in quite precise parameter estimates as a result. Of course, some of the times those parameter estimates will be biased because the model is wrong. And we know from dogma that all models are wrong. It's just a question of how relatively useful they are. Thank you to George Box for the "all models are wrong" quote. So the second thing about the approach in OpenMX to fitting these models is that it does allow for inconsistent intervals between ages at assessment. Third, we're now able, if we have, say, a classical twin study consisting of MZ and DZ twins, we can perform the traditional partition into additive genetic, common environment, dominance genetic, or specific to the individual environmental components. And note that there is also a possibility that what has been assessed as simply residual measurement error, random error associated with each occasion of measurement, we do have the potential to partition that biometrically as well, because it is quite possible that some of the variation in our measures is consistent variation, but that is not associated with the growth processes. So it's possible to have systematic effects which may be shared by members of the same family and detectable as significant A or C covariances across siblings for the residual error. But it just remains an empirical question. We can test the hypothesis whether the residual error really looks like it may be just residual measurement error, or if there's some systematic variance creeping into the residual as well. We may also be able to fit a moderated version of this model that would adjust for different measurement error at different ages. For there to be different measurement error at different ages might sound a rather fanciful thing if one was considering adult populations, but in a developmental population, and especially considering changes in developmentally appropriate language for any type of question and answer test, but even more physical tests that you would expect to be have relatively consistent error, measurement error in height, for example, how hard you press down the thing on your measuring scale, or how hard you pinch the calipers in measuring how thick somebody's skin fold is at the waist, for example. Measurement error of that type might turn out to be systematic. So, for example, a child in a brain MRI scanner might be moving and fidgeting more the younger the child is. And I think this is a reasonably commonly observed phenomenon in neuroimaging studies. So it might be the case that what we've assumed about that measurement error being equal across occasions could be incorrect. And we can consider possible actions to correct that type of problem. So to take it to an empirical case, if we look at the mean thickness of the right cerebral cortex, this was in individuals aged roughly between 8 and 28, there's a notable decrease in mean cortical thickness with age. It goes down from about 35 to 33, so it's not a huge percentage, but it's kind of clearly declining. When we fit the latent growth curve model and allowing for growth over occasions, we can then plot heritability by ages. And here's a plot produced by Eric Schmidt for a paper I co-authored with him about a decade ago. This was based on Jay Gede's data set, a human family study, a health and family study run from the National Institutes of Mental Health in the early 2000s. And this map of the brain, here we can see that same right-hand side of the cerebral cortex. And we look at the heritability, and that's interesting. It's increasing over time. So the red marks more heritable, the blue marks less heritable. And at these 3 selected ages of 5, 10, and 15 years old, you'll notice that the 15-year-old's brain looks appreciably redder and suggesting that there's a higher heritability of cortical thickness at the older age. And the same is generally true when we look at the heritability of the left cerebral cortex. Again, a lot more high heritability in the 15-year-olds than at the younger ages. If, however, we just look at the raw genetic variance component, that is how much additive genetic variance is associated at each of these ages, ta-da, we see the opposite pattern. We see more heritability at the more genetic variance at the younger age and less genetic variance at the older age. So the blue denoting much less genetic variability at ages 10 and 15 is quite striking here. Okay, but that's the opposite pattern to the heritability. What's going on there? Well, the answer to that question is really in the environmental estimates, where we see perhaps a more profound plunge in environmental variance estimates from ages 5 through 15, with an almost entirely blue and, if you go with those color associations, very depressed looking 15-year-olds environmental variance component estimate for age 15. And similarly, if we look at the genetic variance, another thing that we can do with the output from this type of modeling exercise is to put together a movie showing the change in heritability over age. And in this case, it's just a change in genetic variance, which is distinct from the heritability. I really want to rub that point in today. So it starts off with a lot of genetic variance, and especially in the upper areas, but then declines generally to a more green-blue pattern over time. And here we see the other hemisphere doing the same thing. And finally, we can simply plot the mean heritability of over time. But if instead we simply plot the ratio of the genetic to the environmental variance in this fashion, we can see where the counterintuitive result of decreased genetic variance with time, but increasing heritability actually comes from. So both genetic and environmental variability reduce, as does the mean over time for cortical thickness over this age period. And we see that the genetic variance sort of flattens out at around about 3, fairly early on, sort of age 9 or 10, whereas environmental variance continues to decline fairly rapidly during that period and maintains something of a distance from the genetic variance, indicating that you would have higher heritability than environmental variance for all the places where the black line is above the red line, and vice versa for where the red line is above the black. And so there's something like a crossing over of 50% heritability at around 7.5 years of age. So in summary of this brief section, we can estimate ACE model parameters, and we can estimate more than just the A, C, and E traditional parameters. We can go for other variance components, depending on whether our design has more types of relatives. In particular, it's not uncommon to have sibling pairs as well as twin pairs, which enables us to estimate a parameter associated with additional resemblance of twins over and above fraternal siblings. Now, whilst people talk about a sort of special twin environment associated with, something to do with being a twin, we should also remember that because siblings are typically separated in age by a year or more, they are exposed to different secular trends, different, the same, say, national crises or life events are occurring to members of a SIB pair at different developmental stages. Whereas for dizygotic twins, those events are happening at exactly the same ages. And therefore, the special twin environment may at least subsume quite a bit of systematic effects that are due to age times environmental circumstances interactions. One thing I did want to emphasize, of course, was the difference between genetic variance and heritability. I hope that message got through. And the final thing about model fitting generally is understand your data. If you're scratching your head and trying to figure out why you got these particular parameter estimates from an exercise in model fitting, there are usually things in the data that would point the model in that direction, and things in the data that you can inspect to say, oh, is that consistent? Is it the case that the cross-correlation between brain thickness and depression is higher in MZs than it is in DZs, actually giving you a bit of information about potential genetic origin for these individual differences that may not have been so evident from simply looking at one or two age-specific correlations. So oftentimes I recommend going back to the data, and indeed, whilst people typically will do this fairly assiduously, if they're scratching their heads over the results that don't seem to make sense, in practice, I think we should be doing it all the time, regardless of whether the results make sense or not. And that is to have a good look at those summary statistics from the data and see whether those jive with the results that model fitting estimates are putting out. It is very easy to think of model fitting as some kind of mystique and it's magically producing results. Well, it isn't magic. But it isn't an exact science. So optimization sometimes fails, and it's worthwhile making sure as far as possible that it hasn't when it comes to publishing things. And one way of improving your confidence that model fitting results are telling you something sensible about the data is to make sure that they agree with properties of the data that you can simply observe. And often there's no better place to start than some large correlation matrices between the measures or the individuals or both that you're looking at, and either within time or over time. So thank you for your attention. I will pause it there and stop recording somehow. I will shift.